Hello, this is Michael Altos, and we are recording Barash Chapter 12, the Allergic Response, and this is Part 1. First, we're going to have a brief review of some principles of immunology. Just a quick reminder that we have antigens, which are molecules that stimulate the immune response, and these are usually some sort of larger molecule like a polypeptide or a macromolecule. Examples of these might include latex or colloid, volume expanders, and neuromuscular blocking agents. Most of the drugs that we administer are not really complete antigens because they're not large enough, and they fall into the category of what we call haptens. Haptens are small substances that can bind to a larger molecule, and together they can create an antigen. So antibiotics are a great example of a small molecule that in and of itself cannot act as an antigen, but it can be taken up by another protein, and together it can create an allergic reaction. When we talk about immunology, we can divide it into a few different categories. So, for example, we can discuss cell-mediated immunity, in which cells are used by the body to eliminate and destroy pathogens or other undesired cells. And the cells that are responsible for cell-mediated immunity include lymphocytes, which are made up of the many types of T cells and of B cells. T cells influence and implement the immune response, whereas B cells differentiate into plasma cells and are responsible for synthesizing antibodies. Antibodies are immunoglobulins. They are proteins which can recognize or bind to a very specific antigen, and they come in 5 flavors: IgG, IgM, IgE, IgA, and IgD. And when an antigen binds to an immunoglobulin, this activates the immunoglobulin. This sets a whole immunologic cascade into action, whereby lymphocytes proliferate, antibodies are secreted, mast cells degranulate, or complement can be activated. A few trivia points that you may want to keep in mind about cell-mediated immunity. IgM is the largest antibody. IgG is found in the highest serum concentration. Both IgM and IgG are responsible for activating complement. IgG is the antibody that is transferred across the placenta, and it also has the longest half-life of 23 days. And IgE is the antibody most likely to be associated with an allergic reaction, as it is secreted from mast cells and basophils. What are the effector cells in this system? There are monocytes and macrophages, which are responsible for ingesting, processing, and presenting antigens. There are neutrophils that secrete proteases and lysozymes and radicals to kill microbes. Eosinophils, which are responsible for management of parasitic infection and allergic reactions. Mast cells, located in perivascular spaces in the skin, lung, and intestine. They contain IgE receptors and release mediators for hypersensitivity responses. And basophils, which are similar to mast cells in that they are IgE mediated. We can also discuss humoral immunity. Humoral immunity is the component that involves antibodies, complement proteins, cytokines, chemokines, and interleukins. This is in contrast to the cell-mediated component of immunity. All of these components orchestrate cell injury and destruction and are involved in recognition of bacteria and attracting other cells to destroy bacteria. We could also divide immunology into innate and adaptive immunity, where innate immunity is your rapid response team that does not have any specific memory, and your adaptive immunity is more of a delayed time course but has very specific memory of prior exposures. We'll pause here for a moment. If you have any questions on this material, you can go ahead and look them up, or contact me, or bring those questions to class. The other thing I want to discuss in this video is a quick overview of the hypersensitivity reactions. There are 4 of them, and we will outline each of them very briefly. The type 1 reaction, also known as the anaphylactic or the immediate type reaction, occurs when an antigen binds to an IgE antibody, usually found on a mast cell or a basophil, and this leads to a release of vasoactive mediators. Usually a type 1 response requires some sort of prior exposure to the antigen, so that we have IgE antibodies with some memory of this antigen. Onset of the response can be rapid, usually within 5 to 10 minutes after exposure. And clinical manifestations include urticaria, pruritus, laryngeal edema, bronchospasm, hypoxemia, and even possible vascular collapse. In these patients, up to 50% of intravascular fluid may extravasate due to a marked increase in capillary permeability. And management of this clinical condition will be discussed in subsequent videos. In this picture, we see a mast cell with its IgE antibodies attached to it, and antigens bind to these antibodies, leading to the release of vasoactive amines. In a type 2 hypersensitivity reaction, this is considered an antibody-mediated or a cytotoxic reaction. This is mediated by IgM or IgG antibodies, which bind to antigens on the surface of cells, or haptens, which have been absorbed into and are presented on the surface of cells. Here we see an example of complement, leading to IgG, which binds to antigens on a target cell, and this leads to complement activation and lysis of the cell. Or here is another antibody bound to a target cell, and this time instead of complement finding the antibody, a killer T cell finds that antibody, again leading to destruction of the target cell. Examples of type 2 hypersensitivity reactions include ABO incompatibility reactions, drug-induced hemolytic anemia, and heparin-induced thrombocytopenia. In a type 3 hypersensitivity reaction, this is called an immune complex reaction, where circulating antigens and antibodies deposit into the microvasculature. This leads to an activation of complement and recruitment of neutrophils. Here we can see some IgG antibodies. They have bound to the basement membrane of the endothelium. And the IgG antibodies are each attached to antigens. And these antigen-antibody complexes have now been deposited here, attached to the basement membrane. Complement or neutrophils then arrive and further lead to inflammation here. This can lead to a vasculitis or increased capillary permeability. And examples of this kind of reaction include systemic lupus, post-streptococcal glomerulonephritis, and other nephritides and vasculitides and arthritis conditions. Some think that protamine-mediated pulmonary hypertension is also due to a type 3 hypersensitivity reaction. Finally, we have a type 4 reaction, also called a delayed hypersensitivity reaction. This has an onset, as you might expect, of not minutes, but hours, usually 18 to 24 hours, and can last for up to 4 days. In these reactions, lymphocytes bind to specific antigens, and examples of these would include tissue rejection, contact dermatitis, or tuberculosis immunity. We will stop here and pick up with this chapter on the next video.